Hi, my name is Mohammad Hashemian, and I am a PhD candidate working in the David Lab at UC Davis. And my work is titled DNA Damage Detection and Repair Motifs in Mut-YH, Functional Roles of Metal Cofactors and Their Implications in Carcinogenesis. So, a little bit of backstory is that the David Lab studies DNA repair, and particularly the guanine oxidation repair pathway. And what happens to guanine when it's oxidized and forms this very commonly occurring lesion called oxoguanine, or OG for short, shown here as the red O. Oxoguanine is particularly harmful because it can mispair with adenine because of its chemically modified nature. And this oxoguanine A or OGA mispair can form the TA mutation, this TA base pair, going from GC to TA. And so fortunately, we've evolved DNA glycosylases through the base excision repair pathway. And the first one is the human oxoguanine glycosylase in this pathway that can actually cleave the oxoguanine base from the sugar phosphate backbone of DNA, hence why it's a glycosylase. And this results in the formation of an empty site, which can then be filled by downstream repair to give the GC, restore the GC base pair. Now, if you've already formed the OGA lesion, a second enzyme, Mut-YH, which is the topic of my dissertation, can come in and actually cleave out the adenine, the undamaged adenine base, leave behind an empty site to restore the OGC base pair, which gives the OGG1 enzyme a second chance at repair. The problem with Mut-YH is that many individuals harbor mutations in their Mut-YH gene that's inherited or can spontaneously form throughout life, and this can cause a defect in Mut-YH activity and the accumulation of these GC to TA transversion mutations, and that's highly associated with colorectal cancer and generally cancer predisposition, and the best association is with colorectal cancer and predisposition syndrome called Mut-YH associated polyposis, or MAP. So recent advances in our lab have allowed us to map variants, or cancer-associated variants, onto the three-dimensional structure of the protein. So you can see the two-dimensional heat map, where purple is low frequency of mutation and yellow and red is high frequency of mutation. So regions of the protein where there's high frequency of mutation, or these mutational hotspots, surround two of the enzyme's metal cofactors, a 4-iron 4-sulfur cluster, as well as a zinc ion. And so my work has demonstrated that there is allostery not only between the iron-sulfur cluster through these long-range interactions to the active site where chemistry happens, where adenine removal happens, but also allostery from the zinc site to the active site, as well as the zinc site to the iron-sulfur cluster. And I look forward to discussing more about the consequences of these interactions, these allosteric networks, at the poster presentation. And with that, I'd like to thank my group as well as my PI, of course. And especially I'd like to thank the ARCS Foundation. This would be the second time now that I'm awarded the ARCS Fellowship, and I'm incredibly grateful for the opportunities that has given me to be able to travel to symposia and conferences and be able to travel for postdoctoral opportunities. And I'm glad to report that starting in May, I will be doing my postdoc at UC San Diego, where I will continue to do structural biology. And so with that, I look forward to seeing everyone at the symposium, and thank you so much for listening.